So we've got a bonus for today. We're going to be talking about white mold management in really all crops, probably focusing on soybean, but also other kinds of beans. Dr. Marty Chilvers will be presenting, but also Jill Check, one of his grad students, will also be sort of tag teaming with him. And then after that, we'll hear a weather update from our state climatologist, Jeff Andreessen. So I'm going to kick things off by turning things over to Marty Chilvers. Marty, thanks for joining us.
Thank you very much, Eric, for the introduction. Just as we get started here, I do want to thank Michigan Soybean for a lot of support for the different projects we're going to sort of mention today, and also the Michigan Bean Commission, who we also work with. And so as Eric said, Jill Check, a PhD student in my lab, will be joining me this morning. So let's get started talking about white mold. So this is where the pathogen that causes the problem. So Sclerotinia, Sclerotiorum is the name of the pathogen. And here's a really nice example of how important those apothecia or mushroom bodies are in terms of spore discharge. So when there is a change in relative humidity, they will forcefully discharge those spores. And you can see here that they're moving up and away from any boundary air up into the airstream. Most disease is going to really occur locally in patches within a field, but under really severe conditions we can get, or conducive conditions, we can get further dispersal of those spores. And of course, there's three elements to disease. We have to have a susceptible host. Of course, both dry bean and soybean are susceptible hosts, and there's a number of other, especially broadleaf or broadleaf crops that are and weeds that are susceptible to white mold. Have to have that pathogen present. So remembering or making notes of which fields have issues is really important, because those sclerotia can survive in the soil for many years and produce those mushrooms again. And then, of course, the environment. And of course, at this point in the growing season, things aren't looking too good, but things can turn around. So let's talk about a really severe year of white mold. So 2014, we saw a really bad white mold epidemic, particularly through the thumb region here of Michigan. So this particular field was adjusted to 1 bushel per acre. And you can see all the plants there are all lodged because of the white mold. Really nothing left of those, what were good looking beans prior to white mold onset. So why was it so bad? Well, there was a couple of things that happened. Really cool July that is really favorable to infection of the plants, but also moisture. So there are other parts of the country that experience cool conditions, but they lacked the moisture during July. So Wisconsin is a good example. They had cool temps, but they missed out on moisture, where we had above normal precipitation in 2014 during the month of July. And that's what really helped to spur that epidemic. We've done a fair bit of modeling work, and Jill will talk about that in a minute here too. And looking over multiple years, this is a pattern that we see again and again. A cool July and a wet July is when we tend to see most of the bad white mold years. So what can we do to manage white mold? Well, row width and populations are very important, because they affect the microclimate. So that climate that crop is creating underneath the canopy, that can help drive disease. In a minute, buddy. Yep, you can write on that. Thanks. Sorry about that. So the microclimate's really important. And then after that, soybean variety is another really important factor. And then we can start thinking about other active management practices during the year, such as well-timed fungicides. And then, of course, as I've already mentioned, knowing which fields are likely to have disease. And then if we're irrigating, that can also be a risk factor. So we really want to try and minimize irrigation until after flowering, when that major risk period has passed. And just a quick reminder, I think you all know this because it's on the pesticide test, that the main infection mechanism, of this white mold pathogen is through the flowers. So that flowering period is really critical. So here's some data just to support that statement about row spacing. Here we looked at essentially 14-inch versus 30-inch row spacing, and what we saw is about 50 to 60-fold more of those white mold mushrooms under that narrow row versus a 30-inch row spacing. And we saw them develop a little bit earlier. So canopy closure is really critical. That's something that Jill has been continuing to look at as we try and develop new models or improved models for white mold prediction. We do a lot of white mold trial work up the Montcalm Research Farm. We plant most of our trials in 15-inch rows. We also look at row spacing effects, but most of the bulk of our fungicide variety trials are conducted in 15-inch rows. It is irrigated, which is helpful because under conditions like this year, we can push that water to create those microclimate conditions. So here's a slide talking about, there's a couple of different elements to this. It's really demonstrating the huge differences in variety susceptibility to white mold disease and how, of course, that correlates directly to yield. So, and a variety that gets a lot of white mold is obviously going to, get impacted by that white mold and not yield as much as a variety that's got much better resistance. The other thing that we're trying to demonstrate here or show you is the difference that can occur from year to year due to various factors such as field history, and again, the microclimate that you create, the environmental conditions. So in 2022, we had a pretty epic white mold year at that farm, a much greater disease severity than 2021. Due to those different factors. So we, at this location too, we do a lot of fungicide screening, looking for fungicides for their performance of white mold. And we're looking at here a number of different products, just one example trial. We have many different trials that we run, different combinations, different timings to look at optimizing both the chemistry and the timing there to optimize control. Essentially, year in and year out, for us, one of our gold standards there is Endura. That tends to provide the best performance for white mold suppression. But then we have a number of other products there that you can see listed, such as Omega and others. And in blue there is the yield value, and red here we're showing the amount of white mold disease severity. So we'll go through and we'll take those disease severity ratings in early September when white mold is sort of peaking, and then come through obviously with the combine and take yield data to verify those results. So we also look at the various products that are marketed for white mold management, and in this case we looked at Oxidate. We looked at Oxidate 2.0, and in this case, we're looking at 5.0. So this is basically hydrogen peroxide, and it's also got some peroxyacetic acid in there. And in this trial, unfortunately, even 2 doses of that Oxidate actually 3 doses, excuse me, at R1, beginning of flowering, R2, full flower, and R3, beginning of pod set, was no different to our untreated check. We do know that this product can provide disease suppression in other systems. I just think for white mold in soybeans or maybe dry beans, it's a bit of an ask for this product to do a great deal. But we're always sort of looking for those products that make claims or are marketed for white mold management to help provide you the information. And also in this graphic here is our standard sort of test product, Endura. There at R2, providing suppression of disease and much greater yield compared to that untreated check or that Oxidate treatment. So just an example data set there. Some of the other trials we do. All of this work is summarized, and we work with other colleagues, particularly Damon Smith at Wisconsin. He has a pretty strong white mold program. And others around the country. And what we do is we essentially collect this information and put it into a single sheet in terms of fungicide efficacy. And so you can look across this fungicide efficacy sheet and look for the various diseases you might be interested in controlling, and white mold is one of those. And every year we will look at the various fungicide products that are out there and provide you a ranking for those, either excellent, very good, good, or poor if they don't have very good performance. In addition to Endura, we have seen two passes of approach. Lectivar, Omega, and Propulse be reasonable white mold suppression products. And the other thing that I didn't get into a lot today is Cobra, the herbicide. We find that applied at late vegetative stages to be often just as good as Endura under very heavy white mold conditions. And we have plenty of data to support those statements. I'll switch over now to Jill and let her talk about Sporecaster. And just before I switch over, I just want to give you your annual reminder. There's a lot of different mushrooms out there in your fields. And very often folk get confused with bird's nest fungus, which is shown center of screen, that essentially looks like a little tiny nest or an African drum before the skin sort of peels back. That is not the white mold apothecia. White mold apothecia are really hard to scout for, and that's why Jill is working on the projects that she's currently working on. Jill.
Cool. Thanks, Marty. Yeah, so Sporecaster is a tool we use to predict the risk of white mold apothecia in our fields because, like Marty said, they're very small, they're indistinct. It's not really realistic for us to go out and scout our fields for apothecia to try to plan fungicide sprays based on apothecia presence. Next slide, please. This is a screenshot we have from our Sporecaster setup. These are our saved locations, and the pins are labeled with their risk percentage. So across the state, you can see there's varying levels of risk, some higher risk on the west and east coast, and some more medium risk in the center of the state. And that's due to factors like local weather conditions, as well as agronomic factors like whether or not that site is irrigated or dry land, or those rows are 15-inch or 30-inch rows. Next slide. So what I've been working on with Marty is improving the performance of Sporecaster, specifically for irrigated environments. The original model was developed both for dry land and irrigated environments, but after deploying it, we've been monitoring its performance and realized its accuracy isn't really within our target range. So how I am approaching this is I'm using new statistical techniques that I won't really get into, but I think a really big difference will be in this round of model development. We're using data we are collecting on site to capture the effects of irrigation on apothecial development. So we hope that by including factors like how much we're irrigating, how often we're irrigating, and those effects on soil moisture and temperature, as well as soil texture, so really sandy soils versus clay soils at our locations, will help to improve our ability to predict the presence of apothecia. And we're working on that with Dr. Jensuk Dong here. Pictured there is one of his weather stations that he builds and puts out for us to monitor all those on-site conditions. Before we can dive into augmenting the model we already have, we have to first figure out how it is performing as it is now. So what I'm showing here in these graphs is apothecia development through the 2021 growing season. But really the overall takeaway is that the overall model accuracy that year was 76%, and we typically target between 80 to 90% accuracy for these types of tools so they can be really reliable for growers. So we're nearly there, just a little bit off. But where the model really struggled is in what we call timing accuracy, and that is whether or not the model first achieved that high risk threshold the very same time in which apothecia density was high enough to call for a fungicide spray. And that only occurred at our one location in Montcalm in our 30-inch row spacings. So that's the current state of Sporecaster. I have been doing a lot of data collection across the state, looking at when apothecia are developing and where, and hope to have some model improvements to share with you all soon.
Cool. Thank you, Jill. Yeah, Jill's work is really important to improving these models. And I thought some of the information or the data you collected in terms of soil texture was super interesting in terms of, the effect on apothecia timing. So anyway, and just to quickly wrap up, I know Mike gave a talk last week about sprayer setup, things to think about, and there's a link up there in this slide about equipping and operating sprayers. But we want to make sure we've got adequate volume using a ground rig, and we want to try and get canopy penetration here to get that product down into the flowers. And we want to maintain ground speed of 10 miles per hour or less, perhaps. Single flat fan pointed directly straight down into the canopy. We're looking for fine immediate droplets, 200 to 350 microns. And we're using the midpoint of the canopy as a target to try and get those droplets down onto the flowers for optimal efficacy. So in this instance, we're looking for about 12 to 14 inch boom height above the canopy there to target that 17 to 19 inch deposition down onto the flowers. So with that, we'll wrap up. Please put any questions in. Just briefly, other things to be thinking about. We have some trials looking at biologicals, contans, and actually atrazine and metribuzin with Erin Burns, our weed scientist here. And on campus, we developed some white mold because I was irrigating like a madman trying to create diseases, got some white mold. So we actually no-tilled some wheat into that with the idea of keeping those sclerotia on the soil surface, put that wheat in to give it cover, and we also applied contans to try and fix the problem that I created here on our irrigated ground here on campus. So with that, I will hand over to Jeff.
We're going to go ahead and move into our Q&A time. So the first couple questions are going to be about the Sporecaster model. So this could be either for you, Marty, or Jill. Have you seen effects of soil type in your models, and should this be a factor included in the models?
Yeah, that's a really good question. It was a factor we didn't originally consider, but it is one we are now. We are scouting in loamy to sandy soils, and it does look like it is really important. It seems that even in those really wet years and those heavy soils, we see a decrease in apothecia development when things are so insanely wet. So we see this kind of strange response between soil textures, but it is important and we are considering it. So thank you for the question.
Yeah, thanks, Jill. Another question about the model: can Sporecaster be used for all bean crops?
Yes.
Yeah, that's a good question. Yes. So it's a helpful tool, right? It was originally developed for soybeans, but soybeans and dry beans, they have a similar canopy architecture, similar microclimate. So definitely your dry bean grower, it still has some utility to it. The work I'm doing now, I actually work in soybeans, dry beans, and potatoes. So hopefully the tool I will be able to develop can serve all those growers, not just soybean growers.
Yeah, we're working, so Jill and I are working with Jamie Wilbur. Jamie, being our potato and sugar beet specialist, and Jamie actually did her PhD thesis on Sporecaster development, essentially, in Wisconsin. So yeah, we're very interested. Jamie's very interested to see if this can be used in potato as well. So
would that also include sunflower? Is it really any crop that white mold?
Yeah, I mean, yeah, potentially, right? Sunflower's a little bit different because you can get different infection mechanisms, and potatoes a little bit different too. And I'd defer to Jamie on that. But basically, in sunflower, you can get a root infection that's more through mycelium, like mycelium from the sclerotia down in the foot or the root system. And then you get ascospores that infect the flower. The canopy dynamics are going to be pretty different in sunflower compared to, I mean, soybean and dry bean are pretty similar. Potatoes are somewhat similar too. And one of the things we're really interested in, and I think is a really strong driver here, is canopy closure. Like that's really key here. So we did some work actually with shade cloth and set up these boxes, no shade, no apothecia, 100% shade, no apothecia. You've got to have this certain amount of shading to get those apothecia to initiate and form correctly. There's a certain amount of light that's got to filter through the canopy, but not too much.
Thanks. Will the dry soil conditions this spring reduce sclerotia germination, even if we start to get rain during early flowering?
Yeah, I mean, I think it depends. If it's dry right up until flowering, we may not have enough time for apothecia to develop if it was really truly dry. I was just pulling up a few different pubs. Jamie, feel free to chime in as well, but I think it's going to take us at least 2 to 3 weeks from the beginning of moisture to get apothecia formation. So again, in the modeling work that we've done and others, that if we get wet conditions during July, not only is that favorable for infection, but it's clearly also really helpful for that apothecia or mushroom production. So we still have a ways to go if things actually turn around and we get a lot of precip and soil moisture conditions. There's still the risk of apothecia development, but if it stays dry, of course, the risk will be much, much lower.
So how much further can we go? So I haven't seen any flowers yet, but R1 can't be too far around the corner. So at what point can we say, oh, we're past the heat for this year for white mold?
I mean, yeah, I would be waiting until we get flowers. So the period of susceptibility too runs from, beginning of flower to beginning of pod. So we've got about a 2-week window from the beginning of R1. And it's going to depend on the maturity of the beans and everything too. If they're longer maturing window, then that window is longer.
Okay. That is all of the questions that we have in the chat right now, but we've got plenty of time. So folks, feel free to put more questions for Marty and Jill or for Jeff into the chat. Why don't we go ahead? Oh, we just got one dropped in. All right. I think this is another white mold question. What are the similarities and differences between the infection mechanism for soybean and potato that you kind of mentioned? Does white mold infect potatoes through a mechanism other than through the flower?
My understanding is yes, it can infect basically through the soil through myceliogenic germination. We've seen that through in dry beans as well. Doing dry bean root rot sampling, we've actually recovered dry bean plants where clearly the infection is up through the root system, but it's pretty rare in dry beans and soybeans.
Okay. Phil asks, does any effect of plant height on the infection rate of the flowers?
Possibly, where those flowers are placed, perhaps in the canopy. The closer to the ground they are, if conditions are right, then they're going to be closer potentially to spores. But I would say generally not. The bigger thing is, canopy closure. Dense canopies, if you're drilling beans, if you're high plant populations, you're at much, much greater risk than wider row or lower populations.
Yeah, and then very viney growth compared to upright growth is something we see really important. Anecdotally, and at least in potatoes, we've seen be important in infection rate for white mold.
Yeah, and dry beans. Yeah.
Mm-hmm.
I tell you what, I know we have other experts on as well, so we could shift for a second. All the dry weather gets me thinking about insects. I'm not an insect expert, but I certainly start thinking about viruses that might be transmitted as well. I didn't know if Chris had any updates on insects.
Yeah, that's a good segue because during this kind of weather, the sort of insects that are benefited are sucking pests. So you think about things like potato leafhopper, tarnished plant bug, maybe stink bugs a little bit. Soybean aphid is rocking. If you planted soybeans early and they came up in that kind of that early May timeframe, so they're like maybe V4 and they've got that nice juicy trifoliate at the top, and you caught an aphid flight, there's some aphids out there in some of those fields. And And then what else? And spider mite and thrips. We haven't had thrips issue in quite a while, but I've got a few calls. All those things, or most of those things, can transmit viruses as well, especially aphids and some of the thrips and leafhopper vectored things. So I'm putting together FONSFAX emails with all of these pests and a little bit about them, but they're not very common typically. Leafhopper has good thresholds. Aphids have really good thresholds. And if Jeff could get us a little bit of rain, that would really help a lot, because although there's a lot of aphids out there on the tips, so those Mountain Dew colored, juicy little dudes, there's ladybugs out there and there's parasitoids too. So right now there's that battle going on between the dark forces of the aphids and the good forces of the beneficials. What's missing out of the system in this dry weather are those typical fungal pathogens that unknowingly do a lot of control of insects. So Marty doesn't have anything to control, but there's also good beneficial fungi out there that are also being diminished by this weather. And probably the plants are more nutritious if they're kind of drought stressed. So think about sucking things right now. And these are the hardest things to manage. If we get spider mite, it's going to be bad, because spider mite, I would say spider mite sucks because they do suck, but they pierce and they poke and they cause a lot of essentially drying of the leaf. And it's early for this to be happening. So I'll put something together. If you are not on my email list, you can email your email to me, defonso@msu.edu. I'm not sure if the chat saves if people put their email in, but I'm happy to add you to my email blast that'll go out today or tomorrow.
Any updates on potato leafhopper? Have any reports come in of early feeding?
Yeah, they've been here since May, so they've been around. And again, leafhopper is one insect that when you add drought on potato leafhopper, it's not 1 and 1 is 2, it's 1 and 1 is 3. There's some synergistic activity there. But we have good thresholds for potato leafhopper on alfalfa and dry beans. And if you catch it early, they're not hard to kill, they're not resistant to things. So of all the insects, the easiest one to manage in this whole set is potato leafhopper, but it requires knowledge that they're out there and that they're doing something. The hardest one to manage is going to be spider mite, because if you spray, you're on a spray treadmill, and we're not used to that. In Texas, they do this all the time. We don't. And so they're much more difficult thing to manage. They're like my nightmare pest. My nightmare is coming true. So.
All right, Chris, we got a couple of insect questions in the chat. Are aphids on canola the same species that on soybean?
No, the aphids tend to be, there's a few species that are on everything, like black bean aphid gets on everything. Green peach aphid is another one, but the soybean aphid is very host specific. And on canola, I'm not sure what that would be. Sometimes I can identify things from a picture, but I can identify things almost certainly if stuff gets to the diagnostic clinic. I can usually identify those with a host plant. But there's aphids on everything this year. If you go out, you'll see them on grape, and you'll see them on weeds, on the roses. It's just a great aphid, very dry, kind of drought stressed. It's ideal for aphids, but those are not the same species.
Okay. Here's a bug we haven't talked about in a while.
Eric, I need to ask a question real quick before we get off the fungi. Sure. So we're really in a catch-22 when it comes to spraying for fungus and also for the beneficial side of it for soybeans. What do we do there? Which is,
are you talking about white mold specifically? Or are we just spraying just because we want to spray?
Well, I think we have many people will spray because they want to spray.
Mm-hmm.
And if we see that these funguses are killed by fungicide, the beneficials that Chris talked about, Are we more beneficial spraying for insects, spraying for disease, or not spraying and aiding those that are going to be beneficial to the aphids? You know, it's really a catch-22, isn't it?
Well, yeah, from a disease standpoint, we see the greatest chance of return on investment when we're actually spraying to control a disease. I mean, we can get yield bumps, you know, from just spraying, and we don't know exactly why that is, but especially this year, I'd be really concerned about, you know, spiking up insect issues with a fungicide.
Yes.
So I'd be really cautious. Again, just back to white mold really quick. If it does actually turn wet, I would go back to like field histories, where have I had white mold before, and then think about, you know, maybe making applications there if we get canopy closure and the conditions. But just in general, I'd be a little bit more cautious in this sort of a year, just to go out and make a fungicide just because.
Yeah, and this is a system. It all hangs together. So what you do for Marty's disease affects my insects. So you got to think about the whole system. The other thing about aphids, if you planted a little bit later and your beans came up a little bit later, you missed that aphid flight entirely, even if you have treated beans or untreated beans. So you have to go out and look. And a lot of times that fighting that's going on in this system, I often think about early July, you're going to see who's winning here. And there's going to be fields that maybe people don't want to spray because they don't think they're worth spraying. I mean, there's going to be this is going to be a hard thing to work through if it stays dry, because we're going to end up a little bit like 2012 or some of these other dry times where how much are you going to spend on that stand? How much are you going to spend to protect something that the real problem is Jeff, because he hasn't given us rain? If you get rain, a lot of these insect problems are just going to go away, and then we're back to Marty's disease. So we're, you know, it's a system.
Mm-hmm.
Yeah, I've got one more insect question, and then I want to switch to see if there's other specialists on this morning. Colorado potato beetle management. We've got down in our area and in some other irrigated areas, got a lot of potato, volunteer fields, and neighboring field corns, peas. They're seeing adults, egg masses, small larvae. Any comments on Colorado potato beetle management?
So 30 years ago, I worked on potato and only potato, but that was 30 years ago. So I'm not the entomologist for potato now, and I don't know the insecticides have changed dramatically, but I have had this question, and I've tried to kind of parse through the problem is the volunteer potatoes overall. That's just, you know, a commentary that we're not getting winter, or they're not getting killed, or somehow we're getting all these volunteers in here. And then the insects are coming up on those volunteers and then moving. But I honestly don't know what you could do about that. You're going to have to, would you spray a whole cornfield for Colorado potato beetle? And then you'd have to use a product that's labeled on corn. And if you spray early with an insecticide on corn, you could flare mites. I mean, you could be, you know, spraying up spider mite later because you're removing any predators that are there. The thing is, I don't know what kills potato beetle now. And that's something that a potato person would have to know. What do you have? What do you use to kill potato beetles that they die because they're resistant to everything? And what of those active ingredients are then registered on field corn? And that's a tough one.
Yeah. Do we have any other specialists on this morning? Any of our weed specialists on? Forage, irrigation? Anyone else on this morning that has anything to share? All right, I've got no one popping on. I do have one other question that came in about insects. Does soybean seed treatments help with mites and leafhopper control?
So soybean seed treatments are going to last about 30 days from planting, and they will help with aphids, leafhoppers, but we don't usually have leafhopper on soybean, but they would help with leafhopper on dry bean, and they'll help with bean leaf beetle. But those seed treatments are going to wear out pretty quickly here. So if your seed was treated and you planted soybeans early, you probably don't have aphids out there right now. It's only the untreated fields that were planted early. In another couple of weeks, aphids from the first fields are going to then move, and they can colonize now everything because the seed treatments will have run out, if that makes sense. So yes, they help. Then the other thing was mites. No effect on mites. And in fact, seed treatments early may diminish some of the early biocontrol that could have developed, because it removes thrips and things like that. That's prey early for good insects. For all I know, you could actually have a few more mite problems later in treated beans. I know this is complicated, but it's all a system.
Okay, thanks, Chris. All right, I don't have any other questions in the chat. It doesn't sound like we've got any more feedback from specialists. So I want to thank everyone for joining us this morning. We'll close out this morning and hope to see you back here next week.